Hi everyone, welcome to the section of role of free radicals in CNS disorder. By the end of this lecture, you guys will be able to explain the role of free radicals in neurodegenerative diseases, list free radicals causing degenerative diseases, list the sources of free radicals, explain the mechanism of free radical injury, describe the role of free radicals in diseases, classify the antioxidants with example, and discuss the process of oxidative stress response. Let's review the basic concept. First of all, what is biological oxidant? A substance in a redox reaction that accepts an electron is a biological oxidant, right? These include free radicals, reactive oxygen species, reactive nitrogen species. What is a free radical? An atom, molecule, or a fragment of an atom or a molecule that contains at least one unpaired electron in its valence shell and are capable of existing independently. It is highly reactive with short lifespan and has the ability to form new radicals. While doing so, it can damage many cellular components. They are denoted by superscripted dot. They can either donate an electron to or accept an electron from other molecules, therefore behaving as oxidants or reductants, respectively. So the important free radical related to biology are oxygen-centered radicals, which is a subset of reactive oxygen species, and nitrogen-centered molecules, which is a subset of reactive nitrogen species. So you can see that this category of reactive oxygen species and reactive nitrogen species encompass both radical and non-radical compounds. Again, radical species are species that contain at least one unpaired electron in its shell, while non-radical are not free radical per se, but can easily lead to oxidative reaction in living system. This is the list of reactive oxygen species. You can subdivide it further into radical species and non-radical species. And as you can see, the radical species is denoted by superscripted dot. Superscripted dot. This is the superscripted dot. So these include superoxide, hydroxyl radical, peroxyl radical, alkoxyl radical, hydroxyperoxyl radical, and lipid peroxyl radical. Then we have non-radical species in reactive oxygen species as well. These include hydrogen peroxide, hypochlorous acid, hypobromous acid, ozone, singlet oxygen, and lipid peroxide. Next, we have a list of reactive nitrogenous species. These are further divided into radical species and non-radical species. The radical species include nitric oxide, nitrogen dioxide, and non-radical species include nitrous acid, nitrosyl cation, hydroxyl anion, dinitrogen tetraoxide, dinitrogen trioxide, peroxynitride, peroxynitrous acid, nitronium cation, nitrile chloride, and alkoxyl peroxyl nitride. By definition, a free radical is a molecule or atom that possesses an unpaired electron, right? So if an electron is unpaired, the molecule becomes more reactive and unstable than the parent compound. When two radicals meet, their unpaired electron can join to form a pair, and both radicals are lost. However, since most molecules are under physiologic condition, do not have unpaired electron, free radicals abstract an electron from a stable compound, which in turn is transformed into a new radical. Hence, free radicals tend to proceed in a chain reaction. So, potency of a free radical stems from their tendency to be involved in oxidative processes occurring during a chain reaction. And they have three distinct identifiable phases. Very important. The first phase is the initiation, where there is an initial creation of free radical. Usually, it is a hemolytic cleavage with the assistance of heat, UV, and metal catalyst. Next, we have a propagation phase. Free radicals are generated and regenerated repeatedly as a result of chain reaction. Finally, we have termination phase when two free radicals react with each other to form a stable non-radical product. Let's look at the example of reactive oxygen species undergoing the oxidative chain reaction. We have an endogenous or exogenous source producing the initial radical, which is the superoxide radical. This radical is formed by the addition of an electron to an oxygen molecule. This radical is neutralized by superoxide dismutase enzyme in a dismutation reaction, where one radical is oxidized to oxygen while the other is reduced to hydrogen peroxide. As you can see, hydrogen peroxide is not a radical, but it is a part of reactive oxygen species. As it has no unpaired electron, it is a 2-electron product of oxygen reduction. Now, hydrogen peroxide can either form new radical, propagating the chain reaction, or it can form a stable non-radical product, terminating the chain reaction. So let's look at the propagation of the oxidative chain reaction. The new radical could be hydroxyl radical created in the Fenton reaction. This is the Fenton reaction. As a result of interaction between hydrogen peroxide and the metal ions such as iron or copper, which often exist in bound complexes with different proteins such as ferritin or ceruloplasmin. What happens is that under stress condition, excessive superoxide may release these metal ions from their respective complexes. Another way to produce this hydroxyl radical is via the Haber-Weiss reaction. In this reaction, we have the interaction comprising the hydrogen peroxide with superoxide radical. Both can progress in the formation of alkoxyl or peroxyl radicals, which are the products of lipid peroxidation. Next, let's talk about the termination of the oxidative chain reaction. We have two enzymes which can terminate this reaction. One is the catalase and other is the glutathione peroxidase. Now, few things to keep in mind. Glutathione peroxidase is present in cytosol and mitochondria. One thing noteworthy is that it has the higher affinity for hydrogen peroxide compared with catalyst. And glutathione peroxidase reduces and break down not only hydrogen peroxide, but also lipid peroxide by catalyzing a redox reaction with reduced glutathione, which serve as an electron donor. Look at this part of diagram very properly. Okay, so during the glutathione peroxidase catalyzed reaction, we have reduced glutathione, which is converted to oxidized form, which is the disulfide form. This disulfide form has decreased affinity to reduce peroxide. Once oxidized, reduced glutathione can be regenerated from oxidized form by the enzyme glutathione reductase using the NADPH as the electron donor. In the process, NADPH is oxidized to NADP, which is regenerated through pentose phosphate pathway. Let's discuss free radical injury. It is a type of cell damage caused by the formation of free radicals within cells and tissues. The primary targets for free radical reactions are the unsaturated bonds in lipid found in the cellular membrane. Recall that the cell membrane is composed of an asymmetric lipid bilayer with embedded or attached membrane proteins. And one of the characteristics is the fluidity. And the fluidity of membrane changes depending on the composition of bilayer and the environment temperature. The unsaturated fatty acid increases the membrane fluidity. So you can imagine that if there is free radical reaction occurring, it can consequently cause the loss of membrane fluidity, receptor alignment, and potential cellular lysis. Free radical damage to sulfur-containing enzyme can lead to inactivation, cross-linking, and denaturation of the enzyme. What are the sources of free radical? Very important. Both endogenous and exogenous sources can lead to the generation of free radical. Endogenous sources include the immune cell activation, such as oxidative burst of white blood cells. Recall that white blood cells generate free radicals to kill the pathogen. Ischemia can also lead to free radical generation when there is decreased blood flow. Inflammation produces nitric oxide and can cause free radical generation. Redox reaction, such as during normal metabolism, can also generate free radicals. Exogenous sources include the radiation exposure, such as during radiotherapy, example during the treatment of cancer. Drugs such as gentamicin, tacrolimus, bleomycin, and cyclosporin can cause the generation of free radicals, as well as drug metabolism can lead to, during the phase 1, will lead to generation of free radicals. Then we have heavy or transition metals, which can generate free radicals. Now, free radicals can damage to variety of structures, including DNA. It can induce breakage and cause mutation. Cell membrane, it can directly damage the membrane and cause lipid peroxidation, which will ultimately increase the permeability. Increased permeability will increase the intracellular calcium. Now recall that the lipid bilayer is a membrane which is partially permeable, and it controls the intracellular and extracellular environment. We have pumps such as sodium potassium ATPase pump, and we have calcium ATPase pump located in it too. So those pumps also contribute to the regulation of these ions. Since there is an increased permeability, you can expect that there will be an increased intracellular calcium. Now, increased calcium can activate numerous enzymes, and numerous enzymes are activated will damage the cytoskeleton, nuclear chromatin, and ultimately cause the activation of apoptosis, which is the programmed cell death. It is a suicide of the cell. Increased permeability will cause the leakage of cellular protein, which will further cause apoptosis and necrosis. Not only cell membrane is made up of lipid bilayer, but mitochondrial membrane is also made up of lipid bilayer, and lipid peroxidation will occur over there, and formation of transient pore will increase the permeability. Now, mitochondria contain cytochrome C, which will spill out of cell and activate the capsases enzyme. These capsases enzymes is one of the sequential event that activates apoptosis. Again, apoptosis is the programmed cell death. Increased small molecule permeability will draw water inside the cell and cause it to swell, and which will lead to rupture of the cell and apoptosis and necrosis. The free radicals can modify the cellular proteins and can cause microvascular injury as well, which will cause leaky vasculature, ultimately leading to tissue swelling. How do we eliminate free radicals? It occurs via numerous mechanisms. One of them is neutralization by donation of an electron via the antioxidant. Examples are vitamin A, C, and E. Then we have dismutation enzyme, such as glutathione peroxidase, superoxide dismutase, and catalase enzyme. Chelation or sequestration by the metal carrier proteins, transferrin and ceruloplasmin, which hold the iron and copper ions. And finally, the spontaneous decay. What is an antioxidant? So, antioxidants act by delaying or preventing the oxidation of other chemicals. They are usually classified into enzymatic and non-enzymatic antioxidants. However, as you have seen that antioxidants work via different mechanisms, so we can sometimes classify them as primary or chain breaking antioxidants and as secondary or preventive antioxidants. Primary antioxidants actively inhibit oxidation reactions by scavenging the reactive oxygen species and reactive nitrogen species, while secondary antioxidants act indirectly through chelation of the transition metal ions. This table is very important. This is the general classification of antioxidants. Look at here. Antioxidants are classified into enzymatic antioxidants and non-enzymatic antioxidants. Enzymatic antioxidants are further classified into primary enzymes and secondary enzymes. Primary enzymes are the chain breakers, right? Primary enzymes are the chain breakers, while secondary enzymes are the preventers. So, secondary enzymes include glutathione reductase, glucose 6 phosphate dehydrogenase, which is a rate limiting enzyme in those pathway. Whereas primary enzymes include superoxide dismutase, catalase, glutathione peroxidase. These are the chain breakers. Then we have non-enzymatic antioxidants. We can further classify them on their type. Let's suppose we have minerals, including zinc and selenium. We have vitamins. We have carotenoids. We have organosulfur compounds. We have antioxidant cofactors. We have low molecular weight antioxidants. We have polyphenols, which are further classified into flavonoids and polyphenolic acids. So, free radicals play a major role in many neurologic diseases, right? But why is CNS more susceptible to free radical injury? There are multiple reasons. One of the reasons is that the membrane lipids are enriched in polyunsaturated fatty acid side chain. In the brain, this is especially important. The membrane lipids are enriched in polyunsaturated fatty acid side chain. Brain has poor catalase activity with only moderate superoxide dismutase enzyme and glutathione peroxide activity. And finally, some areas of brain are rich in iron, such as globus pallidus, substantia nigra, and circumventricular organs. For this region, CNS is especially prone to free radical injury. So, neurodegenerative diseases are the group of diseases. They are characterized by progressive loss of neurons, typically affecting group of neurons with functional interconnections. Since they are group of diseases, so different diseases tend to involve particular neural systems and therefore have relatively stereotypic presenting signs and symptoms. Let's suppose if the disease involved the hippocampus or associated cortices, patient will present with cognitive changes, often including disturbance of memory, behavior, and language. With time, these progress to dementia. A typical disease associated with it is Alzheimer's. If the disease involves the basal ganglia, patient will present with movement disorders. These may be hypokinetic as seen with Parkinson's disease or hyperkinetic as seen with the Huntington's disease. If the disease involves cerebellum or its input or output circuit, the patient will present with ataxia, example as seen in spinocerebellar ataxias. And if degeneration involves motor system, the disease typically is known as ALS. So, what are the basic findings in neurodegenerative diseases? They are characterized by loss of neurons in gray matter. There will be accumulation of intra or extracellular proteins. If there is cortex degeneration, patient will present with dementia. If brainstem and basal ganglia is involved, then patient will have movement disorders. And the incidence increase with age. The most common denominator among these diseases is the accumulation of protein aggregates, which serves as a histologic hallmarks of specific disorders. So let's talk about Alzheimer's disease. What is Alzheimer's? It is a progressive neurodegenerative disorder and a leading cause of dementia in older adults. It is caused by regeneration of cholinergic neurons, leading to decreased acetylcholine synthesis and impaired cholinergic transmission to the cortex. Most cases are sporadic, while 5 to 20% are familial, and risk factors include age of more than 65 years and genetic factors. Most important thing to note is that it is characterized by two types of neuropathological hallmarks: the senile plaques and intracellular neurofibrillary tangles. To understand the senile plaques, you need to understand the physiology and then pathology. So this is the protein. This is a transmembrane protein, APP, also known as amyloid precursor protein. It has three cleavage sites: one for alpha secretase, one for beta secretase, and one for gamma secretase. So it has three cleavage sites: one for alpha secretase, one for beta secretase, and one for gamma secretase. And with the involvement of cleavage, you can either go into non-amyloidogenic pathway or into the amyloidogenic pathway. So let's discuss the non-amyloidogenic pathway, which is mostly predominated in the physiological conditions. So we have this protein, and the alpha secretase will cleave at its side, and then gamma secretase will cleave at this side, producing three fragments. One is APP alpha, P3 fragment, and AICD. These are soluble fragments and non-neurotoxic. Then we have an amyloidogenic pathway, which predominates in the pathological pathway. And here, instead of alpha secretase enzyme, we have beta secretase cleaving the part of it. Gamma secretase will again cleave it on its original site, producing the AICD fragment along with a beta fragment and then this fragment. This fragment is neurotoxic. It is insoluble and it will start to aggregate and form plaques. Next, we have tau protein. Let's discuss what that protein is. It is also known as neuronal microtubule associated protein tau. Its main function is to promote the microtubule assembly, stabilize microtubules, affect the dynamics of microtubule in neurons, and inhibit apoptosis, particularly in axons. Tau protein normally stabilizes axonal microtubules in cytoskeleton. Hence, it plays a vital role in regulating the morphology of neurons. So, it has more than 30 phosphorylation sites. With the development of tangles, the amyloid tangles, tau protein is abnormally hyperphosphorylated and it loses its ability to bind to microtubules. And the formation of tangles is an important component of Alzheimer's disease, but the mechanism of tangle injury to the neurons remains poorly understood. So there are two pathways that has been suggested. One is that the aggregates will elicit a stress response, which will eventually lead to cell death, or the microtubule stabilizing function is lost, leading to neuronal toxicity and death. Again, same thing. We have this amyloid precursor protein. It has three cleavage sites. This is a transmembrane protein. We have alpha secretase, we have beta secretase site, and we have gamma secretase. In non-amyloidogenic pathway, we have alpha secretase cleaving the protein and gamma secretase cleaving the protein. However, this is non-neurotoxic. In amyloidogenic pathway, we have, instead of alpha, we have beta secretase and gamma secretase. This will produce a beta peptide, which will form monomer, oligomers, and then finally aggregates to form fibrils and plaques. Not only this will form plaques, it will also cause injury to the neurons present aside. It will cause cell death and synaptic dysfunction. Not only that, it will cause the kinase activation, which will cause tau phosphorylation. And since tau phosphorylation, hyperphosphorylation will lead to microtubule disassembly, it will cause neuronal damage. Not only that, this hyperphosphorylated tau will aggregate and form the tangles. So senile plaques are extracellular. They are located in gray matter and they are composed of A beta protein. This is due to enzymatic cleavage of transmembrane ABP by beta secretase and gamma secretase, which will cause A beta peptide aggregation, which is a neurotoxic. Then we have neurofibrillary tangles, which is intracellular, and they are composed of hyperphosphorylated tau protein. Phosphorylation will cause a formation of intracellular fibrils, which is neurotoxic. Next, we have role of inflammation. Both genetic and histologic studies have suggested that innate immunity responds to both of these protein aggregates. So what happens is that the aggregation of these protein will elicit the inflammatory response from microglia and astrocytes, and this response assists in the clearance of aggregate. However, it may also stimulate the secretion of mediators that cause neuronal injury over time, including the free radicals. So the clinical features of Alzheimer's include short-term memory impairment, which converts into long-term memory loss. It is insidious in onset, has slow progression. It involves loss of motor skills, language impairment, temporal and spatial disorientation, and patients are not usually oriented to person, place, time, or events. Impairment of executive functions and judgment, which leads to change in behavior and personality. And finally, in later stages, patient will have focal neurologic deficit. This is a normal brain with cerebral cortex and a healthy neuron. In Alzheimer's brain, you can see the shrinkage of cerebral cortex, shrinkage of hippocampus, and enlarged ventricles, ventromegaly. And on the neuronal level, you can see the plaques and intracellular neurofibrillary tangles. This is the MRI showing the healthy brain and the Alzheimer brain. Ventricles are enlarged and there is cortical atrophy. Next, we have frontotemporal lobar degeneration. It encompasses several disorders that preferentially affect the frontal and or temporal lobes. So they share the clinical features as progressive deterioration of language and changes in personality. Depending on the distribution, if it predominantly involving the frontal region or temporal region, behavioral changes or language problem may dominate. Let's suppose if the frontal is involved, then behavioral changes will dominate, and if temporal region is involved, then language problems will dominate. In general, behavior and language problem precede the memory disturbances. Two pathological subgroups are identified on the basis of histology. Number one is that if the inclusion bodies include the smooth round inclusions of tau protein, also known as big bodies, or if the aggregates are of the DNA and RNA binding protein known as TDP43, then we call it FTLD TDP43. Next, we have Parkinson's disease. It is a progressive neurodegenerative disorder characterized by hypokinetic movement. It is caused by loss of dopaminergic neurons from substantia nigra and elsewhere in the brain. It is characterized by deposition of inclusion bodies, also known as Lewy bodies, of alpha-synuclein in substantia nigra. The alpha-synuclear protein is involved in synaptic transmission, and abnormal protein and organelle clearance due to deficits in autophagy and lysosomal degradation have a role in this disease. So essentially what is happening is that due to defective autophagy, this protein will accumulate and cause the disease. Most cases are sporadic, but point mutation and duplication of the gene encoding the protein cause autosomal form of Parkinson's disease. Clinical features include bradykinesia, which is a slow movement in combination with decreased amplitude speed when moving, resting tremors, cogwheel rigidity, postural instability, shuffling gait. So this is the patient with Parkinson. As you can see, it has mask face, stooped posture, and rigid. He complains of reduced arm swing, resting tremors. If you look at it, he has slightly flexed hips and knees, shuffling gait, flexed elbows and wrists. Next, we have Huntington's disease. It is an autosomal dominant disease, movement disorder associated with degeneration of striatum. Very important. Which part of striatum is involved? The caudate and putamen. Very important question. It is characterized by involuntary jerky movement of all parts of the body. Working movement of the extremities are typical. This disease is progressive, resulting in death in about 15 years of age. Early cognitive symptoms include forgetfulness and thought and affective disorder. There may be progression to severe dementia. And most important thing to remember is that it involves the CAG trinucleotide repeat expansion in gene located on chromosome 4 that encodes for the protein Huntington. Normally, the allele contains 11 to 34 copies of the repeat. However, in disease-containing alleles, the number of repeats increases, sometimes into hundreds. The larger the number of repeats, earlier the onset disease. But once the symptoms appear, the course of illness is not affected by repeated length. Further expansion of CAG repeats occur during spermatogenesis. That means paternal transmission associated with earlier the onset in the next generation. This phenomenon is known as anticipation. Next, we have Friedreich's which is also caused by trinucleotide repeat, except this time it is guanine, adenine, and adenine, GAA repeat in fractin gene. It results in reduction of messenger RNA and protein level of fractin. What is fractin? Fractin is a protein associated with inner mitochondrial membrane, and we know that mitochondria is involved in metabolism and all those important processes. So within mitochondria membrane, fractin is used to move iron and form iron sulfur clusters. Inadequate fractin result in iron accumulation, which leads to degenerative changes in spinal cord, dorsal root ganglia, and cardiovascular system. Finally, we have ALS, which is a motor neuron disease. It is due to death of lower motor neurons in the spinal cord and brainstem, as well as upper motor neurons in the motor cortex. So patient will present with both upper motor neuron and lower motor neuron signs. Most cases are sporadic, but 10% are familial, mostly with autosomal dominant inheritance. Mutation is in superoxide dismutase gene SOD1 on chromosome 21, which accounts for about 20% of the familial forms. Misfold forms of SOD protein will cause the disease and trigger the unfolded protein response, leading to apoptotic death of neurons. However, the most common cause of familial ALS is hexanucleotide repeat expansion on gene chromosome 9. Thank you.